Okay, why don't we get started? Welcome, folks, and to this meeting of the CCGP user group, or whatever we want to call ourselves, the principal investigators and folks from their labs. We're expecting another maybe 20 people to show up for this meeting. It is being recorded, so presumably that's okay with everybody. And I'm Brad Schaefer. I know a lot of you. I probably don't know all of you. I'm the principal investigator and director of the California Conservation Genomics Project, or the CCGP. And what we want to do today is go over primarily sort of where we're at with the project, and with a real opener towards, or a real emphasis, excuse me, on the sort of phase 2 of the empirical work in the project, which is the real heart and soul of the CCGP, which is the whole genome resequencing component of the project. We haven't really started that in earnest, and we will be starting it very soon. So we wanted to try and get everybody up to speed, get everybody kind of thinking in the same way, so that we can do that effectively and in a way that works well for everybody. So with that, let me share my screen. And that's not what I wanted to share, sorry. Let me stop sharing my screen and share my screen again. There it is, okay. There we go. Okay. All righty. There's my screen, and whoops. Hopefully everybody can see this. And so why don't we get started? So what I'm going to do, here's our sort of overall outline today, is for the next, I don't know, 45 minutes or so, I'll talk a little bit about an overview of the project. Presumably this isn't terribly necessary, because if you're on this call, you ought to know what the project is about, but just as a sort of reminder. And then I'll dive in a little more deeply on the resequencing efforts, the whole genome resequencing, the landscape genomics component of the project. I'll then turn it over to Erin Toffelmeyer, our associate director, and she'll talk for a few minutes about where we're at with the reference genome component of the project. And then our hope is that, well short of the first hour, we'll be able to just open it up to questions. And this can be questions, comments. Some of the things we're going to talk about aren't completely set in stone. Many of them are getting close to getting set in stone. But just, if you have questions, if you have comments, if you have additional things we should be thinking about, now is a great time to take advantage of them. And as will be very, very obvious, I hope, or I presume, this is not like a, beautifully rehearsed presentation on my part. I'm telling you about where we're at. We've all been working, our team here has been working very hard to pull these things together. It is absolutely a work in progress. So, sorry, I keep doing that. Okay, so the California Conservation Genomics Project, or CCGP. As you know, it's a collaborative effort to, use genomics to help promote and ensure conservation within the state of California. We're doing that by primarily developing a large, sort of perhaps audaciously large, collection of species of plants and animals, marine, terrestrial, freshwater organisms. that will, we think, help set some new directions and new goals for conservation in the state, looking at a combination of threatened, commercially exploited, and sort of ecologically important species. So the elements of the CCGP, as it stands right now, about 230 species of plants and animals, but that comes out to about 130 projects, right? Because some projects have more than one species. They have a couple of closely related sister species. Looking at 150 individuals per species, creating a high quality reference genome for each of those species groups, not for each species, but for each collection of closely related species. Whole genome resequencing for those 150 individuals for each of those roughly 130 projects. Bringing in GIS, really good, we hope, GIS imagery and data. That's something we will not yet talk about since that's a little bit further down the line. And what we're hoping to do with that is to produce data for management and policy makers at the state and federal levels and for private and public land managers. So private meaning primarily NGOs. So today's goals. I want to talk to you a bit about data flow and sort of goals for those data flows. And this is really kind of an outline of the project in many ways. I'm going to then focus on the whole genome sequencing. I'm not sure why we don't call it whole genome resequencing, but anyhow, it's those 150 individuals. And since we're really launching that part of the CCGP at this point, I want to focus on a number of decisions that our team here, including our scientific executive committee, have been circling around and wrangling with and have come up with as a direction forward to help ensure that whole genome resequencing is done smoothly and efficiently. And then I'll turn it over to Erin, and she'll talk for a few minutes about reference genome progress and updates where we've made more progress. And then, as I said, hopefully have an hour at the end for questions and discussion. Okay, so here is our, we need to get the pictures out of the way. Here is our sort of game plan for the CCGP workflow. And let me just walk you through this, and so that we can see, everybody can see where we're at and where we want to go with this. But before I dive into this for a few minutes, I just want to emphasize, and hopefully this will be clear as I talk us through this image and this workflow, is that at least in my mind, the CCGP it's a collaboration, it's sort of an agreement between all of the PIs who have been funded through it and our sort of central group that's trying to create and deliver a unified project and a unified product to the state of California who funded this project. And what I mean by that is that the state gave us some money. We've distributed it all out to you guys, or most of it out to you guys. And for that, you get funding to work on your favored group of organisms. And that's good. That's a win, hopefully, for the PIs on the project. What you produce, or what you supply, are samples collected in a uniform way. And depending on sort of who does the work, either we do some work and our UC core sequencing facilities create the raw data, or you do some of that work and retain the funding to do it. And then those data are produced. And then the data flow both to the PIs to do whatever they want with it, on whatever time frame they want with it. And they flow to the CCGP kind of central, part of the project. That's the purple on this diagram, to create that product for the state. And this whole thing, if it all works well, it works well for everybody. It works well for the state. It works well for the individual PIs. It works well for conservation in the state. Where it falls apart is if we don't all contribute our bits. And so what we're really trying hard to do at this point here, down here at UCLA, is to create a sort of workflow that's relatively easy so that all the bits will come together. And that's what we really want to talk about here at this point. So, sorry. So the CCGP workflow falls out in a few different pieces. One is the reference genomes. We've talked about this already. About half of you have submitted your material for the reference genomes. This is the one high quality organism or sample that gets shipped to Davis, to Santa Cruz, and to UCLA. Our team and the CCGP, picks up the tab for everything from extraction to PacBio sequencing to OmniC sequencing to RNA extraction in libraries, assembly and posting those reference genomes. And Aaron will talk more about that in a few minutes. The second piece is the whole genome resequencing, or WGS. And this is, as I said, really the heart and soul of the project, and what we're going to mostly talk about today. So this is your 150 individuals that you collect the samples. You can deal with that in a few different ways, and ultimately it will produce 150 individuals resequenced at the whole genome level at 10x coverage. Both of those, the reference genomes and the whole genome resequencing, then go into sort of two different pots. One is this central CCGP repository where the data come in. And we work as a group and we publish, we hope, a couple of really big, really important, really influential papers that cover all of that data and, conceivably have 150 co-authors. And that will push the data through some very, very basic pipelines to produce the maps, the resources, the analyses that we've promised to the state of California. The other thing that happens is that the data goes out to you guys, and it goes to you to do whatever you want with it and that sort of thing. I may have that a little screwed up, but anyhow, that's the basic idea. Okay, so the data flows in sort of two different directions. Okay, sorry. Before I go any further with this, since there are pieces here that need to be discussed, and I will be discussing them in greater detail in a few minutes, I'd like to take a quick break or a quick stop and introduce three different people from our sort of group here, our CCGP team here at UCLA, so that they can just tell you what their roles are in the pieces that are going to follow. So I'm going to stop sharing my screen, and I'm going to ask Tara, Dan, and Courtney to just take a few seconds and introduce themselves to everybody on the call.
So I'm waving in case anybody needs to find my head square. This is me, I'm Tara. So I have been helping so far with the mini core setup. So the mini core is going to be available to those of you who are unable to process your own samples for the whole genome resequencing. The mini core is getting set up so that we can help with that. So far, I've been helping mostly with the physical lab setup. We're setting up a new space specifically for this mini core, helping with planning, cost estimates, feasibility, that type of stuff. In the future, I'll be in more of a help as needed role. All of this, I've corresponded with a lot of you, and in the future, most of that is going to get shunted over to Dan. So that's my blurb.
Thanks. Dan.
Hi, everyone. So I'm over here. You can see me. So I'm new to the CCGP project, and similar to Tara, I will be in the mini core side of things. But I will be more hands-on, so I will be the one who will be processing your samples, receiving your samples, sort of reaching out to PIs and other individuals regarding how samples are going through in terms of the resequencing process. So yeah, you'll probably hear quite a bit from me as you're submitting samples and as your samples are going through the sort of pipeline for extraction and library prep.
Great. And Courtney.
Hello, hi, I'm Courtney. I am the relatively new fund manager / project manager for CCGP. So you'll hear from me regarding money related things and some administrative related things. I do have a background in conservation genomics and worked on a much smaller scaled project similar to this one with multi institutions and multiple taxa. So I can help in a variety of ways regarding those things. So and you'll hear more from me when we do any if you send samples to the mini core and that kind of thing to get all the funding lined.
Okay, great. Thank you very much, you guys. And again, I just wanted to introduce those three people. And I think going forward, it will particularly be Dan and Courtney, because if you use our mini core, or even if you don't, if there's any billing that needs to come back through us in any way, you'll be dealing with Dan on the mini core side of things and Courtney on the billing side. Okay, so let's go through some of the guidelines and ideas that we are developing on the whole genome resequencing side of things. First is just really some, stuff that hopefully everybody knows and remembers from your proposals. And by the way, we take those proposals seriously. So what you said you would do, the sampling you said you would do, we're assuming you're really going to try to stick to that, because that's what the funding went out for. So at the whole genome sequencing, resequencing level, we're, as you know, shooting for around 150 individuals per species. For each of those, one way or another, DNA is going to get extracted, libraries are going to be made and pooled, and they're going to be sent to a core facility for short read sequencing. According to the agreement of our funding from the state, that sequencing has to be done at a UC facility. Right now, we've been working most closely with Davis, Berkeley, and UCLA. If you have an Illumina short read sequencing facility at your campus and you want to use them, that's fine. Basically an S4 lane is an S4 lane, although some facilities seem to be better at producing more data per lane than others. But there are some consequences, some positive consequences that stem from working with one of the facilities that really knows a lot about the program. And we can talk about that. I will talk about that more later. We're aiming for 10x coverage for each of those 150 individuals for their full genome. And just as a thought, remember that organelles, which are somewhat bigger for plants because they've got big mitochondria and big chloroplasts, can eat up a bit of that. And duplicate reads can eat up, sometimes up to 10 to 20% of the reads, not always. So you might sort of keep that in mind as you're sort of targeting more or less 10x coverage. And then my last point is just when you're thinking about the coverage that you want, remember, you buy that in terms of lanes or parts of lanes or gigabases, and sit down with somebody who understands informatics. Maybe everybody on this call does, and that would be great. But if not, make sure you're getting some help so that you're going to be more or less on target. Okay. Again, I'll say this several times. All data, once those sequences are completed, they need to go to two places. They need to go to the lab of the PI who is the lead on that particular project or set of projects, and they need to get over to CCGP. And that's so that we can take all of those data, we can push them through our pipelines, we can take a lead on organizing and developing those big, massive, we hope, hugely important papers that we'll all be a part of. But the data need to get over to us, okay? As a part of that, they need to be made publicly available. Now, obviously, you can't publish sequence data until it's publicly available, at least not with any, most reputable journals. And so the sort of minimum, the maximum, I guess, time, the longest time that the data can remain not publicly available is until the first paper comes out. That paper might come from a PI, in which case they would make the data publicly available. If not, it'll be that joint paper, at which point all the data have to be publicly available. So that's just something that needs to happen, but we need to make sure we do get the data over to us. A few thoughts on timelines. These might be optimistic, but I really hope not. Remember, this is a 3-year project. We're hoping we'll get a 1-year extension, and we're already a 1.5 years into it. So our hope, that we're really shooting hard for, is that all the resequencing for all of those 18,000 genomes should be completed by the end of calendar 2021. All the resequencing data should be shared with the CCGP as it is produced, and therefore no later than the end of 2021. The data, as I said, need to be submitted presumably to the NCBI Short Read Archive, or SRA, and that should be completed by a few months after the data are collected. So we would say, by roughly March of 2022. That submission, for those of you who have done it, it's a little bit of a pain to submit data to the SRA. It's not impossible, but it does take some time. And that is the responsibility of the individual PIs. So the CCGP isn't doing that for all of those data. It's your responsibility to get your data up on the Short Read Archive. We are in the process of setting up an umbrella project. It's called a bio project. And all that is, at least in my mind, it's kind of like a folder for all of the CCGP projects that'll be on the SRA. So it just means that you can go to that and you can see all the projects, all the submissions that are a part of the CCGP. They're all in kind of one place. It's a little bit easier to manage and to think about, since the idea is that this will be a sort of comprehensive collection of data. We just started looking into that. Erin Toffelmeyer, there's her email address, is the one who's organizing that right now, and it's just a matter of getting a bioproject ID. I expect we'll have that in a couple of weeks, although with the holidays, I'm not sure. At which point then, when your data are submitted, they should go in under that bioproject. Okay, so, okay, so what I wanted to talk about for a minute now is submitting that short read data, that whole genome sequencing data, to the CCGP. And we're really thinking about this sort of in two different ways. I'll talk in more detail in a moment about the CCGP mini core that we're setting up and that Dan is leading. If you submit your material to the UCLA, the CCGP mini core for processing, after we've extracted DNA and or made libraries and pooled them, we'll send those to a core facility. More on that in a moment. But as a part of that, we can just say, bill it back to the PI for the sequencing, but include both us and the PI when the data are ready for download. We'll download it from there, and you can download it from there. That's simple and easy. For projects that don't utilize the mini core, and many projects, it's just fine, would like to do that work themselves, then there's a couple of different ways to go. The sort of simplest by far is when you're done creating your libraries, pooling your libraries, getting them to your core, include the CCGP in the submission process. So basically say, as tracking information on your samples goes through your favorite core, when you're told that those samples are available, please tell both the point person at CCGP, that'll be either Dan or Courtney or Tara or Aaron, I'm not quite sure, but we'll figure that out. I guess Aaron, sorry, I can read my notes. You can include Aaron on that, and then when those data are ready, we'll be told, as well as you'll be told. And again, we can just download the data from our side. You can download it to your side. Not all cores allow you to do that. And you should check with your core and see if you can. If you can, it's the easiest for everybody. And then the only other thing you need to do is submit a metadata form. And we have a very extensive, about 10-page document that really outlines everything I'm talking about here in excruciating detail. And that includes a hot link to that metadata submission form. And please use it, okay? If you can't do that, or you don't want to do that for some reason, then what you can do is basically submit the FASTQ files and the metadata after the sequencing has been completed. So you do the sequencing, you download it, and then you'll upload it to an FTP server that should be easy. It might not be fast, but it should be easy to upload the data to us. We're working on setting up that FTP site right now. And please just let us know if that's the situation, okay? And we'll make that happen. Okay, what about before you get your data, when you're thinking about whole genome resequencing, library preparations and sequencing? Sort of what are the options as we see it? And basically, it breaks down into three sort of fundamental categories. One is you can do it yourself, and that's fine. And many labs are very used to extracting DNA and making libraries and pooling libraries themselves. In my own lab, we do it all the time. And that's just fine. The big, and again, there's much more detail on that in that sort of instruction document that we'll be posting probably today. on the CCGP website. The one thing I would say, and we can talk more about this if you want, is that remember to get that really good price, that sort of $7.50 per gigabase sequencing price structure, that is completely dependent on every lane being absolutely full, okay? And that means that it's very unlikely that your project will happen to be exactly one or exactly two or exactly three lanes, okay? That's just a vanishingly small probability. And so what that means is that in order for you to get that best price, and this is again on the whole genome short read stuff, your libraries may need to be pooled with others, okay, from other projects. Now, with Illumina sequencing, that's not a problem. What is a problem is that you can have indexes that conflict with each other. You know, you can have overlap in indices, and then you can't demultiplex. If that is done by a centralized group, whether it's the core facility or our mini core, we'll take care of that. We know what's going in. We'll make sure that the indices get along with each other. And then it can simply be demultiplexed, and you'll pay for the fraction of that lane that you used. Just make sure you've thought about that, okay? You don't have to do it, but if you don't, the price is going to go up, and that's on you, okay? So that's the first thing you can do, is just do it yourself, and that's fine. The second is you can have your core do it. And I think most, not all, but at least some cores offer an option where they will, you know, they'll extract the DNA, they'll make libraries, or they'll just make libraries, they'll pool. It's easy, you just send them stuff, but it tends to be kind of expensive. And that's, again, that cost is on you. You know, we delivered, you know, we funded projects based on a certain fairly minimal cost for this stuff. I think an honest cost, but a minimal cost. And if it's more or less, you know, that's fine, but that's on you. The third option, and the one we're going to talk about some more, is to use the CCGP mini core that we're setting up. And if you do that, then depending on what you send us, we can do extractions, we can make libraries, and we can pull them within and across samples. Okay, so let's say that you decide you want to go with the mini core. What do we need? We need decent samples, okay? You know, we need reasonably high quality tissue or extracted genomic DNA. So you may have already, you know, used this stuff for, I don't know, for doing a RAD study or something like that. If you have that extracted genomic DNA, as long as it's high quality, you can do some quick QCs on it and send it to us and save yourself a little money. Some things, basic things to remember. We can only use this facility that we're setting up for CCGP samples. We can't use it for other even related projects that are very much related to California conservation genomics. I checked with the relevant folks who are funding the core, and they said that that we especially can't use the personnel that we've hired under the CCGP for that, for non-CCGP projects. So they have to be CCGP projects. And we can only bill back for that work. So, you know, you're going to send stuff to the mini core. We're going to take care of it for you, hopefully. We're going to send you a bill. We can only bill back from the same fund that you were allocated originally. So, you know, the money flowed out to you. It's going to flow back to the central facility to cover our expenses. So don't spend up your entire allocation and then say, oh, well, no problem. It's a few $1,000 bucks. I'll just pull it from some other fund. Keep some of it in there to make sure that it can come back to us. Okay, at least that's my understanding of the funding stream. Our pricing. Well, we've been working hard on this, trying to figure it out. Let me just run this through with you quickly. So this is the current pricing that we think we can do and you know, pretty much break even. So if you send tissue, high quality, good tissue to us, and we extract that, extract DNA from that tissue, we make libraries and we pool those libraries, that'll run $30 a sample, okay? If you do the extraction and supply us with good, high quality genomic DNA and it passes muster, that'll save you $10 a sample. So we'll make the libraries and pool them and send them off, and that'll be $20 a sample. Sometimes there may be some additional quality control that we need to do. That adds a few dollars. We may need to do an additional extraction, especially if the material is somewhat degraded or just really difficult to work with. That can add, say, around $6. We may need to do an SPRI cleanup, and if that's needed, that'll be an extra $3 per sample. Okay, so that $30 per sample is what we originally is what I've told people to basically plan on. You know, our numbers varied a little bit during the process of funding all the proposals to between 25 and 35, but 30 seems about right. Let's see, what else? Okay, other important considerations when you're sending it in. For us, plants and slimy invertebrates are the hardest taxa for us to work with. You know, plants are loaded with secondary compounds. They can have tough leaves. They can just be sort of difficult. And slime on invertebrates also tends to sort of, you know, literally and figuratively gum up the works. And those are just sort of difficult for us. That's not to say we won't do them. We may not do them as well as you do them, since we're, you know, the folks who are running this have all worked on vertebrates in the past. We're learning, we're collecting protocols. One thing about it, and you'll see more of this in our instruction sheet, if you have hints or if you have protocols that you really like for your particular group of organisms, please share them with us, okay? And we can, within reason, we can give them a try. Again, this is another way of saying high quality material. Only send fresh material. I know for a lot of plants, herbarium sheets are an attractive option, but they just aren't gonna, all the things our plant colleagues have told us is that they're not gonna produce good material. So please recollect fresh material. For vertebrate people, no formalin preserved material. There are hints that sometimes it can work, but we're not in that game at all. And I said no, and this isn't a real no, but it's a please try not to send so-called hard tissues. And hard can mean difficult, or hard can mean literally hard. So feathers, bones, nails. Those of you who know me know I do a lot of work on turtles, and you know, we have tried endlessly to extract DNA from turtle shells. And it just doesn't work. I mean, it's just, you get lousy, kind of really fragmentary data. And what we want here is wonderful, high quality sequences so that we get those whole genomes at the 10x level across the board. If you do have to send those kinds of materials, A, get in touch with us, B, read what we say on the instruction sheet, but please, if you possibly can, just don't mess with them, okay? For plants, we need at least 1 gram of fresh material, high quality material. Once again, we're just trying to emphasize that. For small invertebrates like insects, we need at least 0.2 of a gram of fresh material, or that's at least our target. And that means sort of material that has DNA in it. Okay, if it's pure chitin, it's not going to really go very far. Whoopsie, sorry. Sorry. For vertebrates, again, about 0.2 grams of fresh material is good, oftentimes cut up into pieces. We have more instructions for that on our document. Should you work with animals that have nucleated red blood cells, you don't need very much. Nucleated red blood cells are a wonderful source of DNA. So that's, 30 microliters is roughly a drop or two. It's about a drop. If you work with mammals, for example, which have anucleated, then you need something more like a ml of blood, okay, just because there's not very much DNA in it. If you're going to skip the extraction part when you send it to us and send us that genomic DNA, depending on the organism and the size of its genome, somewhere between 125 and 500 nanograms. And you'll again see in our instruction sheet, but there's some quality checking that you need to do before you send it to us. Some other important considerations. First of all, please ship, if you're going to ship stuff to the mini core, ship it on a Monday or a Tuesday of a week that doesn't have a holiday. We all know, at least all of us who are in the University of California's know that the University of California mail system, even FedEx, can be, it falls apart on weekends. People aren't around. It just sort of doesn't work. If you ship it on Monday or Tuesday, you ship it overnight, we'll get it Tuesday or Wednesday. That gives us plenty of time in case there's a delay and it has to go an extra day and we don't hit a weekend. I shouldn't have to say this, but ship it legally. If you're going to put dry ice in there, which we oftentimes recommend, declare the dry ice. It's not impossible to ship dry ice, but say what's in there. Because if you don't, there is a possibility that somebody will check and they'll throw it out. And that would be, if it's not, properly labeled, and that would be tragic. Use FedEx. FedEx, especially if you have a university account, is quite inexpensive and it's nice and reliable, and we can track samples. Submit all of your samples for a project at once. Okay, this is a really big one. Our goal is, if you're going to do 150 or 147 or 122 samples, whatever it is, accumulate them, get them all together, send them to us. Okay, that way we can process them all at once. If there's any special protocols we need to deal with, we can deal with them all at once. We can assess the quality of them, which we do for each individual sample all at once. We can do it all at once and have a sense of where we're at, and then we can push all your samples through all at once. Okay, so please don't send piecemeal bits and pieces here and there if you can possibly avoid it. If for some reason you have to, get in touch with us first. What about sample quality? Well, we've gone around and around on this, sort of how to handle this and what's the best way to do it. And here's what we've come up with. Out of your presumably 150, if more than 5 of them on our first pass through look like they're really sketchy, we're just going to stop right then, contact you, figure out what's going on, and figure out what to do about it. Okay. 5 is about 3% of the total sample. That seems like much more than that, and we might want to know what's going on. Okay. If 5 or fewer are lousy or sketchy, we'll just keep going. Okay. If they really look horrible, we'll just drop them and keep moving with the other 145. If they look sort of marginal, but sometimes stuff that looks marginal on a first pass for QC actually works out okay, we'll include them in there and keep going. The reason to drop them is that then you don't have to, your grant doesn't get charged for the sequencing. And if we're not sure, we'll shoot you a quick email, but we want to, we're planning on doing a lot of samples. We want to be able to turn these around quickly. We don't want the queue to be held up waiting for 2 weeks to hear back from you. So if it's just, a few, if it's 2 or 3 of them that look lousy, we'll just make a decision and keep going. Okay. We will send your samples to a core. I've already mentioned that, and it will probably be a UCLA core. We'll do that both because we're hoping to be able to negotiate some good prices, and also, honestly, it's easier. We don't have to be in the business of shipping hundreds of boxes to different cores. We can just walk across the street and have them done. We have 2 cores here at UCLA. Both of them are quite good, and we're going to be working that out, which way to go. So we'll send them to a UCLA or to a core, probably at UCLA, although we're not positive about that, like I say. We'll let you know, and then we'll have the billing on that sequencing, which was a part of your budget, sent to you, and it's your responsibility to pay it. It's also your responsibility to download the data for your own uses, just like it always is. We will also have that data, made available to us. We will download it and we'll push it through the CCGP pipelines. Our goal is to, like most core facilities, we'll keep samples and extracts for 6 months. Assuming everything goes fine, there's no reason to keep those anymore. We will then dispose of them. Okay, so that's what I have to say about sort of the overview, and especially the resequencing and the core sequence, or the core mini core here at UCLA. What I'd now like to do is turn it over to Aaron for a minute to talk about reference genomes and progress and where we're at. And while I'm doing that, contemplate my current favorite CCGP species, the elusive Western spadefoot. Okay, I will stop sharing in a moment, and I will turn this over to Aaron.
Thank you. Okay, let's see if this works. Okay, can you see my screen?
I can sort of see a chunk of your screen. Oh, maybe you just want to be in the reference genome part.
Yeah. So there just should be half of something on the screen right now.
Okay.
So hi everyone, I'm Erin. I think I've met or talked to most of you via email now, so it's good to see everybody again. So I'm just going to give a very quick update on our progress so far in the reference genome. So as you might recall, we sort of launched this phase, the first phase of our data generation in, I think, August. So we're a few months in now. And so just as a quick refresher, just as a quick refresher, our reference genomes are sort of composed of these 3 different pieces. So we're working with PacBio Hi-Fi long reads. So that work's being done at UC Davis, the UC Davis Genome Core. And then we're
Aaron,
also combining that with, yes.
Sorry, you can't quite see the left-hand side of your screen. I don't know, or at least I can't.
Oh, is that?
Maybe it doesn't matter.
Shoot. So can you see like high molecular weight DNA?
I can see
or no?
WDNA.
WDNA, okay.
That's me. I don't know if that's true with everybody or not.
Let me.
Same here.
Okay.
Okay. I will try something else. Sorry about that.
There we go. Yeah. Now we're talking.
Better?
Yep.
Better. Okay. I was trying to be fancy, but I guess I won't be. Okay, so we have our first component, it's the Hi-Fi sequencing done at UC Davis. The second component is OmniC, which is a fancy version of Hi-C. The library preps, the extractions and the library preps are being done at UC Santa Cruz and Beth Shapiro's lab, and the sequencing we'll do at UCLA or UC Berkeley, most likely. And then the third component is generating transcriptomes with RNA-seq. So the CCGP mini core will be doing the lab work for that, and then we'll send those out for sequencing again, either to UCLA or UCB. So that's just sort of refresher overview. And I just wanted to sort of update everybody on the numbers that we have so far and how we've been doing. So In total, we expect to do about 121 genomes. That's our current count, barring any major changes. Actually, we're doing really well in terms of number of tissues submitted for all of the species. So everything in this chart that's not blue is something that's been submitted and is somewhere in the process of data generation, which is great. So we're doing really well for the HiFi and the OmniC. We're about half of all species submitted. We're doing a little less well for the RNA-seq. Oops, sorry about that. A little less well for the RNA-seq. We have only about a quarter of the species have had tissues submitted for them. So especially if you have submitted tissues for your DNA side of your reference genome and you think maybe you can get some RNA-seq tissues, that would be great. Just remember to submit them. And if you need help deciding what kinds of tissues to use or where to send those or any information, feel free to contact me or check out our guidance. We have all the information online right now. And just as a quick reminder, we've said we can do up to 5 different tissues and different individuals, different life stages are great. It's actually better if they're not the individual from your reference genome. A variety of individuals is really useful. So we realize that for a lot of species, especially a lot of these CCG species are threatened and endangered, that might be difficult to get those additional extra tissues. So if you have the option to and you think you can get some, great, let us know and submit them. And I think many, if not most of you, have also been in contact with our reference genome manager, Fringy Richards, about submitting the rest of the tissue. So for HiFi and OmniC, and talking about when you can. And we realized COVID has sort of messed everybody up with their field sampling this year. So, you know, our goal is to sort of get through these as quickly as we can. We realize it's a large data set to get through. So just, you know, do your best to get us tissues and get stuff into the queue. Okay, let's see. In terms of progress and process on our end, so for the HiFi sequencing, we have 16 species that have gone through some part of the process. 2 actually now have completed assemblies, one of which I'll show you in a little bit. On the OmniC side, we have, I think, about 19 in some stage of the process. The workflows for HiFi and OmniC are a little bit different. So HiFi, we sequence sort of one at a time, end to end, and generate them one at a time. For the OmniC, we'll do those more in batches. So we're in the process of collecting enough species to put on a NovaSeq lane to really maximize our efficiency. So we think probably our first deep sequencing run will happen in January, and then we'll have 5 or 6 species completed on the OmniC side. So, and then for RNA-seq, we've got all of our tissues in the freezers right now, and now, actually right now, with the launch of the mini core, we're going to start work on the RNA-seq. And sort of like the OmniC, those will be done in batches, so we can, once we figure out the RNA extractions and figure out how to pool and batch those efficiently, then we'll also be sequencing those in batches. Okay, and then our very optimistic timeline, but I think for the most part we can hit this in terms of reference genome generation. We're hoping to have most, if not all, tissues submitted by July of next year, sort of cover the next spring field season. Hopefully most everybody can collect what they need to, and then we can finish data generation a few months after that, and then finish the assemblies just a little bit after that. So our goal is to be done with reference genome generation, at least the DNA side, by December of 2021. I should say, and I don't have this in here, that for the transcriptomes, we're planning on having NCBI do the annotation. So once we're done with the assemblies on our end, we'll submit those to NCBI along with the RNA-seq data, and it gets put into their annotation pipeline in queue. So we're not quite sure what the timeline for that is, but we'll be doing that as we generate the reference genome assemblies. Okay, and so just as a little bit of a teaser trailer for the rest of the project, here's one of the hi-fi assemblies that was recently completed. So Merle Escalona is a postdoc in Beth Shapiro's lab. She's the one who's running all of our genome assemblies. So if you have genome questions, talk to Merle. So this is for a Western pond turtle, one of Brad's favorite species. And I just wanted to point out that this is an astoundingly good assembly. Our Contake N50s are very high, or sorry, yeah, N50s are very high. This is way higher than even our baseline quality. So we're doing really, really well. And also we have a really nice and complete Busco set. So I think, you know, this is obviously a really good example of what we've been generating. We hope to continue to generate assemblies of this quality. So this is just sort of an exciting thing for us to see that, you know, we're getting really good data, we're generating really nice genomes. And this is even without the OmniC data added to it. So it can only get better, which is very exciting. Okay, so actually, I think that's it for our reference genome update. And we planned now to have a Q&A about mostly the resequencing information. If you also have questions about the reference sequence progress, let us know.
Yeah, okay, great, thanks. Thanks a lot, Erin. It is pretty exciting. I just want to tell you that turtle is actually, some of you may know Brian Todd at UC Davis. It's his, it's an assembly of his. We're sort of partnering with him. The CCGP is partnering a little bit with him on it. And so that set of statistics is from a nucleated red blood cell blood sample collected fresh that immediately went into the core. So, you know, with an N50 of 115 megabases. Remember, our sort of minimum acceptable high quality assembly is 2 megabases. So that's, I think that's right. So this is way over the top. And that's kind of what you can get if you have really good tissue and you treat it really well. So hopefully many of our samples will come in at that kind of level. So at this point, we're right on an hour, so that's great. And what we're hoping is that people have questions. And if you do, I guess we have a few in the chat. And I don't know if we had asked people if they could, if they had questions ahead of time to submit them. To do so. And I don't know if those questions are in the chat or if these are just the ones that were submitted now or how that works. Courtney, where are we at on that?
Yes, I believe there was just for today's session, just one question from that was submitted when someone registered. I'm going to pull that up just now.
Okay, yep. So, you know, I guess I can read the questions in the chat and we can see if we can answer those. And then, you know, if you've got a question, just unmute yourself and ask it after we get through these. Okay, we've certainly got time. Let me read these first, and then I see Neil's got his hand up. You know, we can raise hands or we can just, you know, you can just speak up. So the first one is from Caroline Williams. Maybe I missed this, but when is the UCLA core, mini core, likely to be ready to accept samples? Tara, Dan, what's your thought?
I think our current thought was receiving samples at the very beginning of the new year, just given the UCLA will be closed for 2 weeks towards the end of this month. And just with like any sort of potential complications with receiving these samples. So right at the beginning of the new year, we'll be really taking off and ready to start receiving these samples, is our thinking.
And Dan, let me just ask you, if for whatever reason, you know, somebody's got their full complement of all of their samples ready to go and they've, you know, they've done a little QC on them and they know that they're good. And for some reason, they just really want to get them out the door now. And a few trickled in, would that be okay or not?
I think that would be fine. Yeah, if we could
Okay.
get them in relatively soon before
Yeah.
like the last 2 weeks of the month. Yeah.
Right.
Yeah.
Okay. Okay, great. And we will post the instructions on where to send and what to do and the metadata that we need and everything else. We will get that posted. It still has a few kind of holes in it, but we've all been working pretty hard over the last 3 or 4 days to try and get that cleaned up and in good shape. Aaron's really taken a lead on that. And I think we'll have that posted at the CCGP website today. And remember, for anybody who doesn't remember, Google CCGP space UCLA, and it will take you to the website. We are making a new website, a new updated website that Courtney's the lead on, that will be both a bit more informative and also just an awful lot nicer to work with, or prettier, and more informative. But I think that we're going to wait to release that probably for another few weeks. So CCGP space UCLA, it'll hit it and look around for the for those documents before you send them. Okay. Next is from Jan Vendetti at the Natural History Museum. She says, given COVID samples, are you able to receive tissue samples in December? Well, I think we just talked about that. So I think we can if we have to. Now, and Jan, I'm not sure if you're talking about reference genome samples or
reference.
Reference, okay.
Yeah.
Reference genome samples.
The first,
certainly.
Yeah.
Those can still go in because that we have all kind of set up and ready to go, and everybody's got their freezer spaces allocated. So just remember, ideally you send stuff to all 3 places, to UC Santa Cruz, to UC Davis, and to UCLA. We have an instruction sheet that's been posted and available for some time on that, and my feeling is that there shouldn't be any problem with shipping those samples, but again, I believe it's December 19th, so the end of next week, UC shuts down tight as a drum for 2 weeks. Okay, so don't get anywhere close to that, because who knows what in the world's going to happen on the Wednesday before. Thursday's the new Friday, and Wednesday may become the new Thursday. So who knows? So if you're going to do it relatively soon. Okay. Merle has a note that completed equals contig level assembly. So the kind of thing I think that we just showed for the pond turtle. Jason says to everybody, NCBI is annotating all types of eukaryotic genomes now, or is this animal plant specific? Not sure exactly what the question is there, Jason.
Hey, so your question, it was presented that there wasn't going to be any annotation from the CCGP project, that you were just going to deposit assemblies and then NCBI was going to take care of the rest. But I wasn't sure. That seems different from what I've seen for most of what goes on in the world. So most of the time they don't annotate, they annotate bacteria, and I know there's some animals that are in their special pipelines, but are you actually expecting them to take care of?
Yeah, no, and we've Fringy Richards has who does this kind of thing a lot, has he's not on the call. He has a horrible family emergency in the UK, but he's been in touch with NCBI, and if the genome assembly is of a sufficient quality, and I certainly expect all of ours will be, and if you provide the RNA-seq, they will do it. Now it's just running it through their pipeline, I presume. I don't know if there's much of any human annotation, or curation that goes along with that, but that is my understanding. And Fringy's are, sort of our expert on this, but I think that's all set. Okay. Andrew, do you have a list of species? Do you have a list of which species are where in the reference genome sequencing assembly process? I don't have one handy on the top of my head. We do have, we've assembled a database, and we have a big Excel spreadsheet that lists all of that, and that's that Excel spreadsheet. So again, Fringy Richards, who's sort of behind the scenes largely job, has been to help out with sort of the, as a sort of project manager on the reference genome side of things. Is he's got an Excel spreadsheet that has all the species in it, where they're at in the pipelines. He's getting that information back from the relevant cores and labs. So we do have that.
So just to jump in, we've been talking about this a little bit. You're certainly not the last or the first person to ask this in this last week, actually. So I think our plan right now is to develop just a really simple sort of update spreadsheet on our website, where people can go just to see, like, if mine has made it into the queue and where it is. Probably a little data deficient in terms of how much information you get, but at least that way you'll have sort of a vague update. And then if you need more information from us, you can email us. But yeah, so we'll try to get that set up pretty soon.
Okay.
If you're curious right now, you can send me or Fringy an email, and we'll try to just sort of give you a rough idea of where things are.
Do you think it's worth Aaron posting what we have right now?
Yeah, we could. It's not in a super clean format.
No, I know. I haven't seen it in a few days, but it's not.
But yeah, I think if I took a few days and a few hours and cleaned it up a little bit, we could have just sort of a spreadsheet that's ugly but informational for people.
Okay. Well, we don't want to make it a make work thing, but if a number of people are curious, maybe we can do that. I mean, you saw Aaron's summary, and that's based on that spreadsheet basically. And the bottom line right now is that we're sort of slowly getting that machinery cranked up. I should also tell you, go ahead. What were you going to say, Aaron?
I think you're about to talk about, but I forgot to say during.
Oh, okay. Well, one of the things, we'll see if it's the same thing or not. One of the things that we've done in the last several weeks, I guess, is that so the big bottleneck in terms of time on making those reference genomes is the PacBio sequencing, and the machine that is sort of the darling of the moment on the long read sequencing side from PacBio is called a Sequel 2, or a Sequel 2E is the newest version of it. It's got an extra bell or whistle on it. And we've been doing that work with the core folks over at UC Davis. And we just worked out an arrangement with UC Davis and PacBio such that UC Davis is buying a second Sequel 2E, and we're guaranteeing basically to completely fill it for a year with our reference genomes. So we're guaranteeing to spend the money that PacBio needs to see spent in order for them to be able to give UC Davis a good deal on the machine. Was all signed, sealed, and delivered in a flurry of emails last week, maybe. And that new machine will be delivered either in late December or very early January. Our hope is to have that online, January 5th. That's our hope. We're always hopeful, but it'll be somewhere close to that. And essentially, we'll have the full capacity of that machine for a year. And that's the only reason why we think we can get things done as quickly as we can. And we may have more capacity than that, but that's where we're at. So is that what you were going to mention, Erin?
Mhm.
Yes.
Yeah.
Okay. So that's just a, that doesn't affect any of the PIs one way or the other, except to say that the good news on that for the reference genomes is that there won't be a long queue. There won't, we won't have, there'll be our own internal queue, which is already pretty long because, we've got 60 or 70 samples already in that queue. But it also means that, for example, if the flow cell on one of these machines is called a smart cell, and if it's, say, a couple of real small genomes and they don't fill, each one of them doesn't require its own smart cell, we can sort of intelligently set up runs to potentially combine a couple of small organisms and save the project a bit of money. Or another really important thing is that oftentimes the first smart cell for an organism, there's a little bit of guesswork, and from that you learn a little more so that you can get more usable data out of the next smart cell for that organism. And so for certain things like, one of Jason's spiders needed 4 smart cells, we can line it all up. So you do the first one, you learn, and then you do the next 3. And anyhow, it just gives us a lot of flexibility to hopefully get a better product out at the end of the day. Okay. Anything more on that, Erin? No. Okay. Next question I have is from Neil. We have small individual insects. Neil works on ants, for those of you who don't know. And we'll be doing a chemical extraction beforehand. Is it possible to submit a subset of these to determine if the DNA quality quantity is sufficient? And I will turn that over to some combination of Erin, Tara, and Dan.
Yeah, so for a situation like that, this will be like more laid out in the guidelines once we post them, but we would suggest, if you have the capacity to do so, to do a QC check before you send anything to us. So either doing the gel or the quant beforehand. But if you don't have that capacity, I think we can definitely take a few individuals to sort of see if your chemical extractions are going well. But this will be more clearly laid out in the guidelines.
Yep.
Once we post them.
Okay. And I'm sure that's something that we will see, different versions of, whether it's tiny shoulder band snails or tiny spiders or tiny whatevers. Okay. Matt or Matthew says, can we get the PacBio and OmniC reads as well? My answer to that would be, from a sort of kind of intellectual property rights point of view, sure. From a, I don't honestly know how hard it is, or if it's, I don't know how one transfers that, never having done it before. Anybody have a clue on that?
I would say definitely we haven't set up a system to do that, but we can certainly figure out a way. If you want data right now, we'll have to sort of figure out a way to do it just individually, but eventually we hope to have all the data sort of posted somewhere where it's downloadable and easy to access. So get in touch with us if you have a species that's already been done and you really are itching to see the data. Let me or actually Merle know, or Fringy, and we can work something out.
Yeah. Merle, do you have anything to add to that? Assuming you're
just to say that the idea was to, once the genome was posted on NCBI, the data would also be uploaded to NCBI. So once that's done, you either get it from there, or if you need it beforehand, we just figure out, as Erin said, a way to get the data to you.
Okay. Yeah. So certainly in the kind of short medium run, yes, in the very short run. If you really want it, need it, we can certainly work on that. Let's see, Courtney has passed on from Glenn. What is the least amount of material that would work for the reference genome samples? We lay that out in the guidelines. And Glenn, I'm assuming you're on the line here. You are. What? And I'm sorry. What do you work on?
Well, I'm Glenn Morrison, but I don't recall filing this question. So.
Oh. Okay.
Yeah. Sorry.
So maybe that's not so true. In any case, there's my understanding on this is that there's minimums and there's minimums, right? And there's kind of a minimum where we know it'll work and it'll work well and everybody will be happy and it won't be a huge amount of drama to make it all work. And then there's kind of minimum where it's the rock bottom, scraping the bottom of the barrel. And I think our hope is, just in general, that we can avoid that latter minimum, if at all possible. Doing that oftentimes using really small amounts of data or degraded data. You can clean it up, you can do stuff. We all do that individually in our own labs, but we're really hoping to avoid that if we possibly can. So I think if you have those questions, contact Erin. She's our real point of contact for a lot of this kind of thing. We can try and work it out, but man, read the guidelines that we gave, and if you possibly can, stick to it.
Yeah, and if I can just add, I think that minimum is a little bit dependent on the kind of tissue and the species that you're working with. So if you really think that you're going to be sort of that scraping the bottom of the barrel situation, let me know, or you can also talk to Ruta from UC Davis. She's the point person for doing the high molecular weight extractions for the PacBio side, and then Eric and Sam from UC Santa Cruz are the ones sort of the OmniC team on the Santa Cruz side. Their contacts are all in the reference genome document that I just linked in the chat. So if you're really concerned, talk to them first to try to strategize what would be a good way to approach dealing with your species.
Yeah. Okay. Greg Rether asks if we have, say, 140 of the 150 samples already, but need to do more field work for the remaining 10. Do you want us to wait until we have collected all the samples and submit them all at once? Well, the answer is, why didn't you get the last 10? No, I'm sorry. I think
permits, Brad, permits.
Yes. Greg is in my department. So anyhow, this is an awkward one, but I think the answer is that if, in the case of Greg, those, damselflies that he works on will be back out in the spring in most places, probably. So it's, basically December is shot no matter what. So, if it's going to put things off for 3 or 4 months, and you can round it out and have it all done, I think I would say wait. Okay. If it's going to be another year, then I'm a little less sure, but really we want to try to process them all at once. So I would say wait. I mean, I know that's hard. Certainly for some of the reptiles and amphibians that Ian and I are doing, it's samples are going to trickle in, and ultimately I know we're going to have to make some hard decisions about when to just pull the plug on it and say, okay, we're only getting 142, so be it. But if you can, wait that and get it done, that would be great. But it's a very good question. Okay, that's everything I've got in the chat. Do people have any other questions?
I think we skipped over one.
Did we? I'm sorry.
Yeah, from Matthew Van Dam. Is it possible to post the CCGP's informatics pipelines?
And I think my answer to that is when we have them, it would certainly be possible to post them. I'll just say that we're trying to move all of these different moving pieces of this project through, at a certain level simultaneously. Most of us, certainly I, don't just do this. This is just sort of one project that I do amongst many others in my job. And so we haven't gotten everything done all at once. We figured that those pipelines are something that once we have the way to get the data coming in, then the next step is going to be to hire, we hope, a couple of good, we're sort of thinking probably a couple of good postdocs for a year or so to work both on the informatics pipelines and on the landscape genomics pipelines. I will say we're expecting they're going to be, especially on the landscape genomics side, pretty basic. This is our goal isn't to do super fancy analyses. Our goal is rather to move everything through some basic, informative landscape genetics approaches that will allow us to sort of characterize variation at the landscape level across the state, across species. And certainly when we have those pipelines developed and in place, we will absolutely post them. We also should be soon pulling together a small group of people that can help us develop those pipelines and figure out the best ways to do it and serve as an advisory team for them. So that's the long, short answer. Any other questions?
Brad, I'll just say, yeah, I think actually you kind of answered right there at the end. I was just saying, like, some of us do kind of specialize in informatics. And so it would be great if there was a little bit, I know that you have a team and you've gotten, this is from a leadership there, but if you wanted to open up the kind of two-way street on that part, I think some of us might be able to also contribute there too, or some of our team, if that helps. I know too many cooks are hard, but if it's if there's ways to kind of spread that load, I think we could help. I think.
Okay. All right. Thank you, Jason. Thank you very, very much. Okay. Other questions? Other thoughts? I guess one other thing I will say is that in terms of the mini core, I guess honestly, I at least have a little bit of a sort of it's sort of a mixed feeling about it in some ways. On the one hand, I think if everybody sent their stuff to our mini core, it sort of streamlines things like, pushing the stuff through in a consistent way. It streamlines making sure that we have access to download the short read data and that sort of thing. That's a good thing. The bad thing is that, again, none of us, well, maybe with the possible exception of Dan over the next couple of years, really want to be in the business of running a core. And so if you can do it yourself, that's great, and as long as the other stuff happens, as long as the data stream comes to us so that we can analyze it in terms of, and as long as we all keep in touch on how those data are moving through and it's done in a consistent way, it saves us work and saves us energy. So I think, I don't know, I guess it's just there's sort of two sides to it. And so what we have sort of said is if it would be convenient for you to use the mini core, please, consider doing so, because it will streamline certain aspects of it. If you, and I know a few people have told me, they've got projects on really difficult species or really gnarly ones to get good extracted DNA out of, or their samples, the nature of the endangered species they're working on, most of their samples are really lousy. In that case, certainly, if it's going to take a lot of TLC to get the material through, especially on the extraction side, if you can do it yourself, that's great. Or if you have a special team who you work with, that's great. So there's sort of a balance point there. I guess I'll just sort of mention that. And if you're not quite sure where you stand, get in touch with us. Get in touch with me or Aaron, and we can sort of talk you through it a little bit. Okay. Anybody else? Anybody else? Any questions on this? Okay, I'm not hearing any, and we've been going for an hour and a half, so that's to my mind a perfectly reasonable place to stop. Remember, if there's things you missed, if there's people in your lab who weren't able to attend and thought they were but didn't, or if you, having heard all this, you've got somebody, be they an informatician or a person at the bench who you'd like to hear this stuff, we're going to do it again on Thursday, 3 to 5. I believe I have that correct. And is that right, Courtney?
Yep, Thursday.
Okay, Thursday, 3 to 5. Yeah. Okay. And if you can't make Thursday, 3 to 5, but you'd really like to have someone go through it, or you have questions, or there's something critical that was missed. I'm also, if you send me an email, I'm perfectly willing to sort of do it again, maybe in an abbreviated format on Friday. Okay, but we want as many people, and especially as many PIs as possible, to hear this, just so everybody's hearing the same thing. Everybody's hearing, the same words and has the same understanding. It'll help avoid miscommunications and misunderstandings down the track. So, if again, Thursday, 3 to 5, and if that doesn't work for you, shoot me an email and let me know. Okay, and Carlos asks, will the presentation be available on the website? And the answer is yes. Correct, Courtney?
Absolutely. And I'll just jump in really quick for a quick side note before we start losing folks. Yeah, as we mentioned, we have a new website launching very, very soon with a lot of these helpful information will be also posted to it. So if you received a request from me to send a photo and maybe a blurb about your species, let me know if you can send that, or if I'll keep an eye out for any that are incoming. So when we do launch it, we can have everybody showcased there. So, and that also on the same, in that same aspect, if you don't see it on the current site, it's because it's on the new one more than likely if you've already sent it to me. Also, just quickly, since we'll have a space for sharing any, we have the means to share some updates from any of the collaborators. So if you have any fun lab updates or, ways for people to get involved, I can also, post something like once a week to showcase your lab. That's all.
Okay, and then I have one other thing that I'll mention since we do have a couple of minutes, which is on the reference genome side of things. So, we're going to be producing a lot of reference genomes. Right now, I think Aaron said the head count was 121, and we're looking at perhaps another 10 pest species that the California Department of Food and Ag, we're collaborating with them on. So it might get us up around 130. And what we are hoping, and what we're in the final stages of negotiating, is that for each one of those reference genomes, you've probably all seen these really short little reference genome release papers. There's not much in the way of interesting science in them, but it's just a way that as your reference genome gets posted, it's an article, it's a formal peer-reviewed part of the scientific literature that people can use to cite your genome if they're using it for anything, your reference genome. And the Journal of Heredity, which is the flagship journal of the American Genetic Association, is a journal that many folks associated with the CCGP have worked with quite closely. They've just started a new section in the Journal of Heredity, which is specifically for this purpose. It's just a very short, sort of template-based, quick and easy publication. And what we are really, really hoping is that all 130 of ours will be published as 130 individual papers through the Journal of Heredity. There's no absolute requirement for that, but it's something that we're negotiating an open access, very reduced open access fee for those papers. And it's just a way to get, again, it doesn't in any way, shouldn't in any way steal any thunder from a bigger, fancier release or a comparative one or anything like that. It's just a way to say that as your genomes are posted, that if people start using them, and in many ways we hope that they do start using those reference genomes, that they cite you, and therefore you get credit for that work. And so we'll be reaching out to everybody about that. We're in the final stages of negotiating that open access fee with the Journal of Heredity. I'm hoping we can get it down to a sufficiently low level that the CCGP, from the funds we've kept aside and haven't pushed out to you guys yet, that we can just pay for it all. I'm working on that. I was working on that early this morning. So anyhow, that's just something which I hope is something positive to look forward to. These are papers that will, should almost write themselves. I mean, they're, as I say, really quick. They're 1,000, 1,500 words, and there's a template for them if you go to the Journal of Heredity website that's publicly available that you can take a look at to get an idea. We're thinking about modifying it a little bit for the CCGP. So that's just a thought for everybody. And if anyone has questions on that, I mean, you'll certainly hear more about it from us all, or from me in the near future as we work out the details. But we're hoping that that'll be a win for everybody, and that and you can bring on obviously whoever you want as co-authors on it, but presumably the person or the lab who's primarily responsible for that species would be the lead author on it, and just something that everybody can do. Okay, I'm not seeing any other questions. So given that, thanks very much for being here. And remember, you've got sort of a resource team at this point. Aaron is very knowledgeable about everything from from soup to nuts in the project. You can always contact me. Dan's going to be our lead on the mini core and all things associated with it. Tara is our real technical lead on the mini core and other issues. Courtney, both on the financial side and also on the sort of project management side, things like the website and that kind of thing. Okay, so any questions? As always, let us know. Shoot me an email, or give me a call on my cell phone if it's something you don't want to email about, and we'll work it through. Okay. All right, folks, I think that's it. So look forward to hopefully a good break, and then just opening the fire hose of samples to get sequenced. That'll just be great. Okay. All right. Talk to everybody later. Bye-bye.